Okay. All right, we're recording now.
Hi.
Okay. Moving on. Oh, so all our membranes of all the different organelles organs or tissues could be different and are different because of the proteins. Where's my clicker?
That's a good question.
I don't know, so like does the recording like show the screen?
Yeah. So
Oh,
when you
lit.
see the video, you see this, whatever, right on here, and then here up. All right.
Hey Cameron.
Or whoever in the world decides to watch this video.
Hi, person.
So how are membranes permeable to things that are hydrophilic? It has to do with the proteins that are in the membrane. So your neurons are, especially in your brain, your central nervous system, they are a lot less permeable to things than the rest of your body is because of something called the blood-brain barrier. If you took psychology, you probably learned about it, or anatomy. We'll talk about it later. But the unique collection of proteins will allow the cell to have to be permeable to different substances, but also do different stuff. And we'll talk about a lot of functions that proteins can have besides being channels for diffusion or active transport. Anyways, the semi-permeability is because of the proteins there. So, for example, here, what do you think that picture is supposed to indicate to us?
That they have to go through a certain protein component.
Yes, yes. These channels are actually very specific. There's not just one channel that allows everything in and out. So each channel protein is specific to a certain solute. Okay. So, and even the amino acid one, there would be a different channel for like every single different amino acid, 20 of them. There's not just one channel for all amino acids. There's not just one channel for all sugars. There's a glucose channel, and then there would be like a fructose channel or whatever, sucrose channel, bless you. They would all be different. Okay. So I'm going to skip through some of this because you don't really need to know it. Now, I want you to think about that question. Just think about it. Don't answer it yet. Think about all the macromolecules. Huh? Think about all the macromolecules and then think about why proteins. Why not other lipids? Why not nucleic acids? Why not carbohydrates? Oh, there it is. Thank you. Why proteins? They can fold.
You got us Kerr.
Wait, what? What are we doing? What are you showing me? It's annoying. They can't hear me when I'm teaching and recording at the same time. You're saying, are you good? Yeah. The little light turns red when it's sharp. Are you good? You sharpen out? The light turns red. All right. What? They can fold.
Sharpen and then she broke it.
Well,
Yeah.
there's another.
They could be like polar.
What's the fault? Your problem, not mine.
What Max said, think about it. There's 20 different amino acids. Some of them are non-polar. Where would these be in the membrane? The non-polar ones, where would they be? Where would they be? The non-polar ones, like if this purple thing is a polypeptide, where would the non-polar amino acids be located? Come like draw it, circle it. Wait, what do you mean? Just like show where you think they would be on that protein. Yeah, throw it. On the protein. It's not what I'm saying.
This one's not.
I
know it's
Okay. So those non-polar amino acids would be located in the hydrophobic core. Where would the polar or the hydrophilic ones be?
Oh, the one that's on the side.
I'm circling.
Oh my God.
Eddie Young.
Okay, that's, well, the protein is purple.
Oh.
The circle on the circle protein.
Right here?
What?
What are you asking?
Where are the hydrophilic arteries?
Where is it?
Please, come circle. Sit down. It's all right.
I bet it's obvious too. It'll be mad.
You are. The whole part is purple. There.
Are you?
And then the other part.
Wow.
No, don't. Oh, his circle?
Yeah.
Nah, I mean, part of the answer. Yeah, I was about to say. I gave a fourth of that answer.
Okay.
It's still a piece.
The R groups that are hydrophilic would be on the aqueous environment inside and outside of the cell. They would be in one other place. Does anybody know where that place would be?
Yeah.
So think about it. This is our protein channel, okay? And like my hands are the membrane. So the hydrophilic ones would be like up here and down here, but they would also be in there, in here, right? The hydrophobic R groups would be where it touches like the phosphoryl tails, right? Makes sense?
Wait, I'm just looking
In the middle of the channel. If this is a channel and something that's hydrophilic has to pass through the channel, then the R groups that are on the inside of the channel have to be hydrophilic as well. Does that make sense? So proteins are the perfect molecule to make these channels with because they can be both nonpolar and polar at the same time. And they can fold in these unique conformations to make things that are like channels. Make sense? Yeah. They're all in aqueous environments. If they completely dehydrate, completely, they would die. So that's how they die. Yeah.
No.
Alright, so here are the nonpolar ones. Let's see your questions. And then the hydrophilic ones. Yay, let's move on. So, the amino acids that are located within the membrane, and then the part that actually faces the tail would be the nonpolar ones, right? And they're hydrophobic, and that's why it could exist in there. It would hold it in place. And on the outer surfaces of the membrane and on the inner part of the channel that like touches the extracellular fluid, it would have to be the hydrophilic ones, not necessarily polar, but the hydrophilic ones. Make sense? Okay. So I'm going to kind of skip. We've talked about aquaforms before. I'll come back to this later. Okay. So the proteins the unique selection of proteins in the membrane will help determine that membrane's function, what can diffuse in and out of it, but also what type of tissue it is, what type of cell it is, and what it can do. That's sort of how we talked about it. We've talked about peripheral proteins, and we talked about the transmembrane proteins. I'm going to kind of move on. So all the different functions that membrane proteins can have. They're not just channels. They can have a lot of different functions. Now, the ones that function as channels only function as channels. They won't do multiple things, but what I'm saying is different proteins can have different functions. So we focus in this chapter really on transport, whether it's passive transport or active transport. And we'll talk about some of these other ones in other chapters, but I do want to mention them now. So some proteins and some membranes can function as enzymes and can catalyze specific cell reactions. Some can function in what we call signal transduction. So a lot of times, ligand, here's a vocab word. Have we talked about a ligand before? Write this down. A ligand is a small signaling molecule. I'll talk about ligands a lot. I don't want you to be like, what is she talking about? A ligand is a small signaling molecule. here is a ligand. It looks like an orange piece of pizza. This signaling molecule is sort of communication thing. It needs to tell this cell, hey, you need to divide. To be like a growth factor. But that signaling molecule, if it's hydrophilic, it can't get into the cell. So what does it do? It basically binds with a protein in the membrane, causes that protein to change its shape. That protein will basically tell another protein, which tells another one, which tells another one, which tells another one. It's sort of like playing telephone. Until the cell gets the information that, hey, we need to do all the stuff we need to do to divide. And then it does what they need to do. We do all this in chapter 11. Anyway, signal transduction. Cell recognition. So in that video we watched yesterday with the macrophage rolling along the cell that lines the capillary, remember it's bound, like a protein on there, bound to another protein on another cell? That would be cell recognition. A lot of times glycoproteins are involved with this, so these proteins that have carbohydrate chains attached. Sometimes proteins help to anchor other cells to each other and make them stick together. The cells that make up your skin, your integument, these cells are tightly joined together. You wouldn't want them not tightly joined together, and then you would just rub on your build and come off. So there's a genetic disorder called epidermolysis bullosa that happens.
No, like the way you just said all of them, like there was maybe 2 hours left. Because
Epidermolysis bullosa. Have you heard of it before?
I feel like I've seen like videos on Twitter, like they have to stay like patched up all the time.
Yeah.
Like it's bad.
Yeah.
How long do they live?
They can live for a long time. It's just pretty painful.
She's gonna start in one gut show. Watch.
Google will show you like the worst pictures of it ever. And then it'll show you the average ones when you get further down. I hate it when it does that.
That's not a bad picture. But your skin basically blisters a lot. A lot. It's a genetic disorder. Most EB, they call it EB. Most EB cases are genetic. Usually it's autosomal recessive, meaning you just have 2 recessive alleles. So it's super painful. Let's see. I don't know. Close your eyes if you get grossed out. Wait, I'm going to image it.
Yep, there it is. I knew
Yeah.
it.
Yeah. See, look, the baby. Poor thing.
Can't even land the blanket.
Oh, it's so sad. Anyways, okay.
No, I have
Bumpy layer of skin.
Okay, I had it like this.
The bumpy layer of skin under your skin?
Yeah.
Well, you have a hypodermis that's made up of fat. Is that what you're talking about?
Okay, well, I mean, I don't know what it is, but I got burned before, and like, you know how they blisters, there's like a really big bubbly, and then under it, there's like a weird.
Oh, well, it could be your dermis. You have an epidermis, and your dermis, usually when you get a blister, it's usually your epidermis has come off of your dermis, and it's fluid filled. And under that, like if it comes off, your epidermis has no neurons or anything in it. It's all basically kind of dead. So when it comes up, they tell you to like leave that in place, like try not to pop it, because if you do, all of a sudden now your dermis is highly innervated, has lots of neurons. So then your dermis is exposed with lots of pain and all that anyway. Yeah. Yeah, it hurts. Okay, anyways. And then these proteins can also function to attach to the filaments of the cytoskeleton to help maintain cell structure and support, and also filaments of the extracellular matrix. We'll talk about that later. So they can have lots of different functions. Like I said, we're going to really focus on transport, but I'm going to also take a minute to talk about glycoproteins and glycolipids. Yeah.
Okay, so for these self-collections,
Mhm.
they see the glycoprotein, and then the other proteins are releasing.
It's attached to it. It binds to it.
Okay, it looks like it. Okay.
No, yeah, it's binding to it. Yeah. All right, so I'm going to take a minute and talk about HIV. We talk about HIV a lot in this class because it's a virus. We have a whole chapter on viruses, and it affects your immune system. I have a whole chapter on that. So we talk about it a lot, and I'm going to go ahead and introduce it now. HIV stands for what? Yes.
That is AIDS?
No, that is not AIDS. HIV stands
It
for,
leads to AIDS.
well, it can. It doesn't necessarily, yeah.
Oh, you know what immunodeficiency? Yeah, that's what I'm saying. Like,
Human immunodeficiency virus, it can lead to acquired immunodeficiency syndrome, which is AIDS. It doesn't necessarily, Magic Johnson does not have AIDS, he has HIV.
you don't have AIDS.
No. No, yeah. So some people, it depends on the type. There's lots of types of HIV. Some are more virulent than others. Virulent, if it's a very virulent form, it can cause and would cause AIDS much more quickly. Once you develop AIDS, your immune system is basically non-functional and you can get sick. And most people that die of AIDS die of what we call opportunistic infections. They die of like pneumonia or really rare infections that normal people don't get because your immune system works. They can die of the flu, stuff like that. Or they die of really random rare cancers that most people wouldn't get. Anyway, but they will develop that quickly. Some people have a very non-virulent form of HIV where it could take many, many years to develop a, anyway, we'll talk all about that when we do our immune system chapter. But what I wanted to mention,
He had AIDS though, right?
I don't know.
You don't know who that is? You know who that is?
I mean, I've heard the name. I feel like he might have been in a movie I saw.
Okay, it was probably straight out of Compton, but you never heard of
Yeah.
In the
Yeah. Okay. Yeah. Yeah.
Yeah, he had AIDS.
Yeah. Yes. Okay. Yes, I remember now. I know. What? Yeah, no, sure. Time to get to me. All right. So, anyways, what HIV does is what it actually affects. Now, this is a macrophage, but you have a lot of different types of white blood cells in your body. One type of white blood cell is not a macrophage, but it's called a helper T cell. Helper T cells are the targets for HIV. HIV has a particular protein that will bind to a protein on your helper T cells that is called a CD4 protein. So here's the CD4 protein receptor. And then there's an associated, what we call co-receptor called CCR5. And in order for HIV to infect your helper T cells, both of these have to be there and have to be normal conformation or normal structure. So HIV binds to it, and once that happens, what it causes is basically the whole phospholipid, we call viral envelope. Not all viruses have them, but HIV does. It will fuse with the cell membrane of your helper T cells. You can see that here, dumping the genetic material into the helper T cells, and at that point on, it does its thing and can make your helper T cells start making more HIV viruses and not doing its job.
Like, for HIV, like, if you don't have that co-receptor, then it just, like, doesn't, stay in your system, because it's already in your body, but, like, it doesn't.
It will not be able to replicate itself, so eventually would die out.
Oh, okay.
I mean, viruses aren't alive, but it would, yeah. So some people are genetically, they are unable to get HIV because they have a mutated either CDR protein or CCR5 co-receptor. And so they will not be able to get the HIV virus because it won't be able to dock to your helper T cells and it won't infect them. Now, the crazy thing about HIV is it mutates really rapidly because it's called a retrovirus. And we'll talk about why that is later on. But you can be infected with one form of HIV, but within like a year have like a totally mutated version of it because it mutates so rapidly. Anyway, are you going to say something? No. Okay. We'll come back to this stuff later because, like I said, it's a big part of our virus chapter and of our immune system chapter. So you don't need to know this stuff. You need to know the cell membrane, we say, is like a fluid mosaic model. So fluid means the stuff moves around. The phospholipids can move. They can actually switch places with like their neighbors and rotate laterally. They usually hardly ever flip-flop or switch places with the one underneath it. It would require too much room. And the proteins move around. It's all very fluid. We call it a mosaic because it's composed of a lot of different stuff, right? Okay. You've already talked about this. Oh, so let's talk about glycolipids and glycoproteins really fast. And I feel like I've mentioned this before. But these glycolipids and glycoproteins, major function is for cell recognition. It's like your ID on your body or at school, okay? If your immune system is trolling along and it sees a red blood cell, okay, that has normal glycolipids or glycoproteins on it, it recognizes it as self and it completely ignores it. It just goes away. This is a macrophage. They're phagocytic, so they extend all these pseudopodia and eat stuff. So these are supposed to be just pseudopodia. I didn't make it. I don't know. Anyway, so, but these glycolipids and glycoproteins are like the cellular IDs, and you have your very own unique cellular ID that is unique to you, and specifically you. No one else in the world will have that unless you have an identical twin, right? But if for some reason you ever get a blood transplant or blood, what's it called? Transfusion, and they give you the wrong blood type, okay? If you are type A and they give you type B, I hope that never happens to you guys. But your white blood cells will run around and be like, whoa, you are non-self. Like when you're not wearing your ID and like Dr. Silvernail, like, young man. So what happens is that then it starts attacking and basically it causes your blood to all clot up and then you can die. So that's the function of these glycolipids and glycoproteins, self versus non-self. And it's the basis of what we call immune rejection. If you ever get an organ transplant, then you would be put on immunosuppressant drugs for a long time, if not the rest of your life, so your body doesn't reject that organ. Because it would reject it because there's no one in the world that's exactly like you. Now, they try to find a really close match to match as many things as possible, but they can't get an exact match unless they, one, find an identical twin, or they can grow your own, which is technology that eventually will be available. Yes.
I don't know what else to ask.
I'm sorry.
What's the most rare blood type?
I think it's AB, maybe negative, maybe positive.
I thought I had a rare one.
Positive, probably.
Oh.
Colin, what's your question?
Oh, I was gonna say, is there at least 7 people in the world that would like that?
I've heard that before. There used to be a video, something called Killer on Campus. It's about meningitis that Pablo used to show like many years ago. It was like a VHS, like old. But there, at the beginning of the tape, there was a girl that like walks along and she looked exactly like me. It's freaky. Like really weird. Huh? No. Yeah, right. It was weird though. Really weird. I tried to find it online, but you can't find it anymore.
I remember the show.
Anyways, okay, we'll talk about this later. I wanted to remind you of something. When we talk about these proteins that are like, how do the integral proteins, how are they actually synthesized? Remember I showed you this picture yesterday? So look, here's an integral protein right here, one of these transmembrane proteins. Well, where it's synthesized, first of all, do you remember where it would have been made and inserted into the membrane? In this whole process, where would it have been made? In the rough ER. That's where the synthesis would start. And instead of being shunted all the way inside the lumen, it would just stop the synthesis so it's stuck in the membrane. And it would travel from the ER to the Golgi, get modified if it needs to get modified, and then travel eventually to the membrane and would fuse. And then there's our membrane protein with the little oligosaccharide facing out to serve as a glycoprotein now. See that? Okay. But notice that where it was made, it would be synthesized and looks like it's inside out on the inside of the cell. I mentioned this yesterday. I just want to kind of point it out to you there. Okay. Good. We're not finished. This PowerPoint is 84 slides. Okay.
What slide are we on?
Not very many. Probably 30 something.
28, I think.
29.
29.
Ah,
Yeah.
nice. That was close.
We got a ways to go. All right. So remember, the second law of thermodynamics is that everything in the universe tends towards entropy or disorder. So imagine I dumped an entire can of Lysol right here. What would happen to it? The molecules.
Spread.
They would spread until eventually what would happen?
Everyone knows spread.
Everyone was
Ran out of room to spread, or another way to say that would be it reaches equilibrium. Yes. And you can say dynamics because that means that they don't actually stop moving, they still move, but then that doesn't move. All right. So imagine this situation. We would say, here's some dye molecules. This would be a semi-permeable membrane that is permeable to the dye in this case. And here's water. There's water on both sides, but no dye over here. So what direction does this dye move?
Right.
from the left to the right, like so, and it would continue to move until it reaches a state of equilibrium such that the dye molecules are equal concentration on both sides of that membrane. Do they stop moving, period?
No.
No, but we say the net movement is zero. So if one moves on one side, another one will sort of replace it. They still move, but the net movement is zero. I also want to point out this is called diffusion. Diffusion is the movement of something, anything, from an area of high concentration to an area of low concentration. But I also want to mention the rate of diffusion. The rate of diffusion is directly proportional to the concentration gradient. So which one of these two has a greater concentration gradient? This one? Let's say A. It's like the eye doctor. A or B?
B, or, sorry.
You know when they do that, eventually you're like, I don't know, there's a
Looking at the
A.
same thing.
I'll just pick A. Huh? Because the things are like super small differences, and sometimes it can make a big difference, and the test for astigmatism. So, what I mean by the concentration gradient is sort of like a hill. Okay, so imagine A, like you're on a really steep hill. So if I put a ball up here, it's going to roll down pretty fast, right? Compare A hill to B hill over here. This hill would not be as steep. Okay, so it'd roll down, but it just wouldn't get to the bottom as quickly. So the rate of diffusion, your hill is going to be a lot steeper the more different either side is. Does that make sense?
Yeah.
Okay. Does somebody say no? The concentration gradient will directly affect the rate of diffusion. So there's a higher gradient here, difference between this side and this side versus here. See that? When I mean gradient, like you can think of it like a hill. The greater the difference between the two sides, the steeper the hill. Does that make sense now? Yeah. So there's not as big of a difference here as there is here. So it won't diffuse as quickly now. Yeah. Okay, so simple diffusion is just from high to low. We call this passive transport because there's no energy needed for it. We'll talk about water later, so skip. Another thing you want to know, or you need to know, is that solutes will diffuse down their own concentration gradient. So even though there's like more molecules over here versus here, if this membrane is permeable to both of these, the purple will diffuse down from high to low purple, and the orange will diffuse down high to low orange. So the purple in this case will diffuse that way, and the orange will diffuse this way until they reach their own equilibrium. Does that make sense? Good. Questions? Yeah. Well, the purple would probably diffuse faster than the orange because this is a much different gradient than this would be. Does that make sense? Like there's no purple over here and there's like 12 over here. I don't really know. And then there's no orange over here, but there's only like 5. No, as long as the membrane is permeable to both of them. When you start making the membrane not permeable to some, it's going to totally change things. We'll talk about that tomorrow. Probably.
So wait, but like the purple one finished before the orange, or with the orange still?
As diffusion goes on, the rate will change over time.
Yeah, but I mean, so would they keep going?
Good. I forgot that one moved. I don't know, because it would slow down proportionally.
All right.
I feel like the orange would happen before the purple would.
Yeah.
Because there's not too many. Okay, we've already kind of talked about this stuff. So, yep. All right, one thing we need to know, we've already talked about, is that lipids will diffuse down or through or out of a membrane with no help. Like they don't need a protein channel because they're hydrophobic. Okay, so we say the diffusion, anything that's hydrophobic, is truly passive transport or simple diffusion. Okay? No energy needed, down a gradient with no protein helping it. Called simple diffusion. That's what lipids or anything that's hydrophobic would do. Oxygen gas. is non-polar, so it would do the same thing. Say that again. I'm defining the word simple diffusion, which is up here. Simple diffusion is down a concentration gradient. The things are non-polar or hydrophobic, no protein needed. I think that's all. So in this case, what direction would these lipids diffuse? Into the cell or out of the cell? Into the cell. Good. Now, the cell is permeable to a lot more than lipids, and it has to do with the fact that there's proteins permeating the membrane. There's either these holes or these channels. So diffusion through a protein channel is called facilitated diffusion. Facilitated diffusion directly means diffusion through a protein channel. The protein is the facilitator. Facilitator, yes.
And something like this, stuck in the membrane.
In between, what do you mean in between?
Say that whatever is going in between right now just stops in the middle.
Well, usually not. I mean, I guess it could, but I feel like it would only do that if something was wrong with the protein. And that protein wouldn't, I mean, unless it got unstuck, it wouldn't function anymore. But the cell could make another one and invert it. So, not. Okay, I don't know how to store it. Yeah.
Like,
I hear it.
anything that's non-polar, so it seems to go right through.
Yeah, anything that's non-polar does that. Just like goes through.
So like, can it stay in between the?
No, it wouldn't.
Yes, you know?
It wouldn't hang out there. It would just keep going.
So how exactly, like, I figure we'll talk about this when we talk about viruses, but how do they get through the membrane?
So like HIV, it has a phospholipid. A lot of animal viruses have like it's called an envelope, a viral envelope. It's made out of phospholipids. It's basically a cell membrane, kind of. So that would just fuse with the animal cell membrane.
All right, so
Dropping.
just accept it like nothing else, or there's no like.
It would have to fuse or dock with a protein already on our membrane. Does that make sense?
Okay, so it'd have to use just a random protein that's already there.
Well, it wouldn't be random, but
Or-
yes.
Yes.
Yes. Yes.
Okay.
Okay, so I think how lipids would go through like the hydrophobic core, but like how did they diffuse through the polar head?
Well, they don't. The lipids aren't ever really in solution. They're kind of just in suspension all the time, anyway. So, like, how do steroids travel in your bloodstream? They're in an athlete's environment, traveling around. So they're not. We wouldn't say they're dissolved. They're just sort of in suspension. Does that make sense? That's kind of why they do that. They're just there. They're just not impeded by that hydrophobic core, like something that would be hydrophilic would.
So what if like on the outside of the cell, there are only like 5 of them? Or would the,
No, it's still diffusion, it just wouldn't be a fast rate of diffusion, because there's 4 right here.
so like what is just like switching?
I mean, diffusion, they would go from an area of high to low. So, right, if it became high on the inside, it would just diffuse out.
And then
Make sense?
I do.
Yeah. Okay. There's different types of protein channels that facilitate diffusion is with a protein, okay, with a facilitator. So there's different types of protein channels. This one is sort of like just an open channel all the time. Now, what's interesting is that these protein channels, now some of them don't look like that. Some can look like, hold on. Go away. Some can look like this. Do you see this picture here? Where they can close, okay? So they can be, what's interesting, and there's a whole slide on this here, but I'm just gonna kind of say it all. But they can be inhibited. So something can bind to the protein channel, causing it to change its shape and close it, okay? Just like an enzyme. They can also become saturated, just like an enzyme. So imagine the doorway to my room, like unlimited number of people can't go in and out at the same time. It can only accommodate so many people. Same thing with these protein channels. Only so many solutes can go in or out at any given time. So they can become saturated, just like an enzyme. There we go. So they can be regulated and all that. Okay. We're going to talk about facilitated diffusion. Some can have a conformational change and can flip-flop in one shape versus another, and that's just what that is indicating in that picture. Okay. So there's two types of other channels I want to mention. They're called ion-gated channels and voltage-gated channels. And I'm going to talk about them using my nervous system PowerPoint. If you've never taken anatomy, this might be a little bit intimidating. Don't freak out. We'll get to this later. But there are some things you need to know. So this is a neuron. Have you seen this before? Maybe in like 7th grade. I hope you've seen this before. So a neuron in general has some sort of cell body where all the organelles are located. These things coming off of it are dendrites. That's where ligands are received and bind to them. A neuron sends a signal in the form of an electrical signal, a change in charge. So the electrical signal will be sent down something called an axon. It will terminate in something called the synapse, and that electrical signal is converted into a chemical signal called a neurotransmitter. Taking AP psychology, you learn this as well too. Okay. In general, most animal cells, just ignore this stuff, are charged. Right? This. Okay, ignore this. But a lot of our cells are charged, not just our neurons. Some plant cells can do this too. This is something called membrane potential. When there's a charge difference across the cell membrane, you need to know this. The membrane potential is developed usually by something called the sodium-potassium pump, which we'll get to in a little bit. But that energy difference, or the charge difference, is sort of like energy in a battery. Tyler. See, there's Fletcher, go away. All these kids are on fall break, and they are going to come by. Oh, she just texted me and said she wants to come tomorrow. It's Friday. I won't be here Friday, but
I don't think any of us will be here Friday.
you won't. I'll be at R&E all day working. But
It's not for me though.
yeah, I heard. And the sunlight just didn't.
Oh, yeah, that was weird. She was playing on her phone.
I emailed about her.
She was having some stuff.
She won't come back to my room.
I thought she was chill.
Oh wait, the
She
lady.
let a student that wasn't supposed to be in here for like 30 whole minutes. That kid could have been, I know Sanjay, I love Sanjay, he would never do anything wrong. But the fact that that happened is like not good.
Oh yeah, that person.
I think it was.
I wanted to ask her, like,
I know, but that's not good. Like, no.
I left my Chromebook
What is
in I left my Chromebook in the van room. I had to grab it.
Sometimes people here are so psychotic about rules, but then one day, it was last year, some kid from Blythewood just like walked up here in the middle of lunch.
It was Westwood.
Well, Westwood happened one day too. Westwood was, yeah, he did too another day. Yeah. But no.
Yeah, like I went to just grab my Chromebook and like she was on the phone the whole time. Like I need to ask.
Oh. Anyway, sorry, we're recording. Anyway, so this membrane potential, it's like energy in a battery. A battery, has positive and negative charge, and as the ions or the electrons travel, energy can be used. Anyway, same thing here. So our neurons in particular are charged, and we say they're polarized. Okay. Now, when a membrane receives a signal, remember I talked about a ligand? Okay. A ligand will bind. Imagine there is a protein channel, like right here. Okay. This is a sodium channel, and this little ligand bound to it. Here's our ligand. And when it bound, it opened it up. And now sodium ions can rush in. Yay! We call this a ligand gated ion channel. Yeah. Maybe the other class we had that conversation. All right. A ligand gated ion channel will open when a ligand or signaling molecule binds to it. And here, a neurotransmitter or something binds to the neuron, causing the sodium channel to open and sodium rushes in. When that happens, what happens to the charge? Notice what happens to the charge. It flips, right? We say the membrane becomes depolarized. That's the beginning of our electrical signal that has to send all the way down the neuron. The problem is on our big neuron, imagine our big neuron, our ligand is only released up here. It does not release all along the way. So how does this signal move all the way down the end of the neuron? That's because neighboring to these channels are other channels that are not ligand-gated ion channels. They're called voltage-gated. And voltage-gated ion channels open when the voltage changes around them. So this voltage is now changing, which will then open voltage-gated channels nearby, then allowing sodium to rush in down here.
This makes so much more sense now that it's explained from biology.
Huh?
From like biology, this makes so much more sense because we didn't really talk about the ligands in biology. So like it makes so much
Good.
sense.
I'm glad.
Just, oh yeah, it opens up. Like, lets it down.
Yes, no. So that's how the signal moves down. So there's voltage-gated channels all along the axon of a neuron. Same thing on a muscle cell. Remember you learned muscle is the same thing. So those are the different types of channels. And we'll come back to this when we do chapter 48 or 49. But I just wanted to mention that when we go to, remember this from anatomy, Jillian? Yeah. We might have gone through it really quickly. Huh? Yeah, this is the same PowerPoint I used with y'all. Here's my picture. There. All right, so here's again a picture of our ligand-gated channel. Ligand bound, opened up, sodium ions can rush in. Ligand goes away, sodium channel closes, they can't get through. Voltage-gated channel is the same except for its voltage changing, not the little signal molecule. So, okay. Now, the next thing we need to talk about is something called active transport. And this is when molecules or ions need to move up their concentration gradients or against their concentration gradients. You need to go from a low to a high. It's like if I put 100 people in this room and I want to put 5 more. Like, whoa, okay? So it takes energy to do it, and they're going not where they want to go at all. It's not something that's going to happen naturally. Diffusion happens naturally. Active transport does not. So we say the proteins that do active transports are pumps, sodium, potassium pumps, proton pumps. We call them a pump. And they use the energy of ATP. And I'll talk about how ATP is actually used. It doesn't just blow up and burst everything. No, it phosphorylates stuff, remember. The example I'm going to use is something called the sodium-potassium pump. Okay, now I'm just going to use this picture and talk about it with the picture, then I'm going to show you an animation. There's a better image in your book. It's not as confusing, but just follow with me here. The sodium-potassium pump is in basically your membranes of all your cells. It's in a lot of organisms. This picture, the way it is organized, looks like it's one cell with like 6 different proteins. This is the same protein at 6 different stages in the process. Okay, so I'm going to start here. So this is like stage 1, stage 2, stage 3, stage 4. It's not one whole cell. So let's just start here. So the sodium-potassium pump, what it does in general is it pumps 3 sodium ions out of a cell, and it brings 2 potassium ions into the cell. You take $3 out of your bank account, but only put $2 back in. What happens over time to your money? You go negative, right? So over time, what happens to the inside of your cell membrane? It becomes negatively charged. The sodium-potassium pump helps to generate that membrane potential I mentioned ago. But what does it do? How does it do it? So this protein channel flip-flops from 2 different conformations. So in this conformation or shape, it stays towards the inside of the cell. There are 3 binding sites in it for sodium ions. When the 3rd sodium ion binds to the binding site. Now keep in mind these ions aren't looking to bind. They're not like, I'm going to sodium-potassium pump. They're just floating around randomly inside the cytosol. When it happens to bump into that, it'll bind to it. All right. So the concentration of sodium will directly sort of influence how fast these pumps work. Because if there's not a lot, it might not open and close very quickly. Anyways, when the 3rd one binds, it causes an ATP molecule to phosphorylate it, add a phosphate group to it. Remember, the way ATP works is by making things unstable and more reactive. So by phosphorylating this protein, what it does is it causes it to change its shape or change its conformation such that it flip-flops. Notice it goes from here to here. It changes shape, okay? Whenever a phosphate is attached to this protein, it's in its conformation such that it's facing the outside of the cell. So phosphate is in all these pictures and it's faced outside. It's not on these and it's on the inside. Anyway, I also want you to look here when it's in this conformation such that sodium can bind. The sodium binding sites are nice and big and will accept the sodium. When it changes its shape and flip-flops, those binding sites now have changed shape dramatically. You see that? In such a way that it won't hold the sodium anymore and it kicks them out. Physically, it kicks them out. So they leave, and now the potassium binding sites are nice and ready for the potassium. In this other shape, they weren't. Here they are. Potassium will come in. When the second one binds, it causes the phosphate to be kicked off. Then it flip-flops back to its original shape with no phosphate attached. The potassiums leave, and then it starts all over again. I'm going to show you an animation. I sent you an email this morning with all these animations here. For a lot of stuff we talk about, and I'll use these a lot as we go through the year. But the one I'm doing right now is this one.
The sodium-potassium pump is an active transport mechanism that is driven by the breakdown of ATP and works through a series of conformational changes in a transmembrane protein. 3 sodium ions bind to the cytoplasmic side of the protein, causing the protein to change its conformation. In its new conformation, the molecule becomes phosphorylated at the expense of a molecule of ATP. The phosphorylation induces a second conformational change that translocates the 3 sodium ions across the membrane. In this new conformation, the protein has a low affinity for sodium ions, and the 3 bound sodium ions dissociate from the protein and diffuse into the extracellular fluid.
I didn't know that they caused.
The new conformation has a high affinity for potassium ions, 2 of which In this new conformation, the protein has a low affinity for sodium ions, and the 3 bound sodium ions dissociate from the protein and diffuse into the extracellular fluid. The new conformation has a high affinity for potassium ions, 2 of which bind to the extracellular side of the protein. The bound phosphate now dissociates, and the protein reverts to its original conformation, exposing the 2 potassium ions to the cytoplasm on the inside of the cell. This conformation has a low affinity for potassium ions, so the 2 bound potassium ions dissociate from the protein and diffuse into the interior of the cell.
That help? Maybe? All right, well, you have them all. Let's move on. Yay, okay. Well, I told you there's a lot of notes today, I'm sorry. Keep going. So this is just a review of all the transports. I'm going to show a picture. I'm here to talk about it. This is something like in your book. So again, simple diffusion is for anything that's hydrophobic, goes right on through the membrane, down its gradient. No ATP needed. Facilitated diffusion is using a protein facilitator, again down a gradient, no ATP needed. Active transport uses a protein, but also uses ATP and moves molecules up their gradient. Cells will use active transport a lot to get stuff from the environment that might be rare, like sugars. It needs as much sugar as possible, so if it's not very common in the environment, it could use that. Or particular ions, or something like that. Anyway. Let's move on. We already talked about this. Oh, I do want to mention the proton pump. Proton pumps are active transporter, but they in particular move protons or hydrogen ions. A lot of times they will pump them outside of the cell. This happens a lot in plant cells. Again, it contributes to membrane potential because you're taking positive out, but it's used also to develop a hydrogen ion gradient. Plant cells in particular use this to then allow hydrogen ions to diffuse down their gradient through something called a symporter or co-transporter, and with it brings sucrose up its gradient. So it's like using it for active transport without actively transporting the sucrose itself. I think this is in your book. Anyways, it's how plant cells hoard or get lots of sucrose in them. Yeah.
So how's it transport the lipids?
Mm-hmm.
Is there any way to stop it for the cell to
No.
stop it? So it just happens?
No, which is why, like, for example, your blood-brain barrier, anything that's like hydrophobic will just pass right on through it. Like, your blood-brain barrier is way more specific than anywhere else in your body to help prevent certain toxins and things from getting into your brain and killing you, but it's totally unable to affect things that are hydrophobic.
Okay.
So, yeah. So no, you can't stop it. There's no way to stop it. Yes. I don't know what this says. Let's see if it even works. Oh,
Wow.
of course. Okay, let's move on. And talk about big stuff. So your book talks about exocytosis, endocytosis, and something called receptor-mediated endocytosis. You might have ignored this page. It is the last page of the chapter, and it looks a little different, and I feel like sometimes you see those, and you're like, I don't need to know that. You do need to know that. It's on page 138, right here. Okay, I don't know if you read it or not. You need to know this, and so I'm going to talk about it now. And there's a specific example of receptor-mediated endocytosis that I talked about that I don't think is in your book. So let's talk about it. So how about things that are super large, like big polypeptides that just can't possibly fit through a protein channel? Cells might be able to take them in, not need to take them in, or cells might need to secrete them, get rid of them. They might make them. So anyway, so endocytosis and exocytosis and receptor mediated. All of these use vesicles for transport. So in this particular picture, this is showing exocytosis. So this cell has large quantities of a particular protein, let's say it wants to secrete into the bloodstream. Packages them in a vesicle. The vesicle travels on a microtubule monorail using motor proteins and takes it to the cell membrane. It fuses and drops out these proteins to go wherever it needs to go in the body. It takes ATP to do this because the motor proteins need to walk. So all this takes ATP. All of these things take a lot of ATP to do. And then there's phagocytosis and endocytosis. So let me talk about these. This first one is called phagocytosis. Only animal cells do this. First of all, because plant cells have a cell wall and they can't extend pseudocardia, but also plant cells don't need to eat because they make their own food. Phagocytosis is when the cell sends out these cellular extensions called pseudocardia, surrounds some sort of food particle, produces a food vacuole. What does this food vacuole eventually do inside the cell? Fuses with a lysosome for digestion, right? Good. Phagocytosis is what we call sort of cellular drinking. So the cell will invaginate. So the cell membrane like kind of comes in and produces a little vesicle, and it's non-specific. It takes in whatever might happen to be in the fluid surrounding the cell at that particular time. So in this case, it took in one purple and like 4 of these little green dots, whatever those are. Non-specific. Cellular drinking is what it's called. Okay. Now, receptor-mediated endocytosis is what we're talking about on the bottom. And this is a way for the cell to be a little bit more specific in what it takes in. So in this case, there are receptors. These little things look like Y that will bind the little purple guys. Notice the little purple guys are kind of rare in the environment. You see that versus the green? But if the cell wants to take in more of the purple, but doesn't really care about the green, it can utilize something like this. And so when enough of these little receptors have bound to the purple little guys, it'll cause the cell membrane right there to invaginate and form a vesicle. These little things look like black hairs or something called clathrin. It's a protein, and the proteins are associated with these receptor proteins, and when they've all bound a ligand, it will cause this whole thing to undergo a process that will invaginate and form a little vesicle. The receptor-mediated endocytosis allows for the cell to bring in specific ligands or molecules from the environment. We do this particularly to take cholesterol out of our bloodstream. We use receptor-mediated endocytosis to take cholesterol out of our bloodstream. So we have little receptors that will bind cholesterol. And that's how we take it out. Why is it bad to have large quantities of particularly LDL in your bloodstream all the time? Remember I told you about what it could do? Let's see, does anybody know what it could do?
Isn't it like
Sort of, yeah.
isn't that supposed to happen?
No,
In bulk?
why not? That's what I'm asking you.
Because why is it?
Why is high cholesterol bad?
Oh, it can clog up the red blood cells.
Okay, now you're talking about the clotting. Yeah, you're right. But what is it? Okay, so it has a tendency to accumulate in the walls of your arteries. I feel like I've talked about this. Not even the other class.
Is it called?
Atherosclerosis. Yeah. Remember? Anyways, and then when that happens, your vessels narrow, and when they narrow, that's when the clotting can become a problem. So you weren't wrong. So we need to take it out of our bloodstream to try to prevent that from happening. Well, there are some people that have a genetic disorder called familial hypercholesterolemia. You have this whole PowerPoint. And this is not in your book, but I want you to know it. This. They have a genetic disorder where their little receptor proteins are non-functional. They can't take the cholesterol out of their bloodstream. Because they can't do it, it stays in there all the time. They can develop heart disease, atherosclerosis, hypertension at a very young age compared to normal individuals, like teenage, 20s, where it's supposed to be normally, it's like, 50s, 60s. So, anyway. There we go. This is all, like I said, I emailed this to you. Just want to mention a real life example of this. So, create. Start on this. I just want to start on this. Might as well. All right. So, tomorrow, we're going to do the rest of this, but we're also going to do those problems, and I have some other stuff we're going to do. I won't be doing those all period tomorrow, I promise. I hate it. So we have to talk about osmosis. So we talk about water separately because water is so different. And of all the stuff we talk about in this chapter, the water part is the most important part. It's the basis of our big lab we're doing this week and everything. So
How big is that lab?
the next one is going to be a 2-page lab report. It'll be like 2 1-page lab reports. There's 2 parts to the lab. It won't be a long one. I'm not making you do long ones for every single one.
Oh, okay.
So don't. It won't be like the last one.
I was like, I don't know if I can take another 12.
No, I remember that.
All right, so osmosis is the diffusion of water. We talked about the diffusion of water separately because it's so important to survival of cells. Something I didn't mention. So the way water diffuses is just like anything else, but water diffuses from an area of high water concentration to an area of low water concentration. So now I want to point you or make you look at my little things. These are called YouTubes. These used to not be funny until there's something called YouTube now. But this is called a YouTube apparatus. In this YouTube apparatus, okay, there's a semi-permeable membrane at the bottom separating this side from this side. But in this case, this membrane is not permeable to those purple dots. It is permeable to water. So what happens to the water?
It'll go towards
It will diffuse towards the side that has more solute, and then this will happen over time. Now, look. So what's going on? Let's talk about the water concentration. Do you see how it looks like there's more water over here compared to here? Yeah, you see that? Now, how does it actually happen? If anything is a solute, now we're not talking about lipids here, but if it's a solute and it's dissolved in water, okay, it is either polar or it's charged. So remember what water does to things that are polar or charged. It bonds, it hydrogen bonds to them. Forms these things called hydration shells. Y'all remember that? No? Yeah? Hydration shell. Hold on. I don't know if I put it in here. Hold on. We talked about it before. Let me find a picture. I'm sorry, I know this is a lot. Like this. Okay, so this is showing an ion. So this is a really large negatively charged chlorine ion. You see how the water, like the hydrogens, will associate so they're around it? It'll completely surround it. Not only, this is 2D, but like it'll go in front of it and behind it. When the water is surrounding it, hydrogen bonding it, that's called a hydration shell. Okay? Anything that is polar, like your proteins, the water will associate different ways and put in the hydration shell. The water molecules that are involved in the hydration shell are what we call bound water. They're not likely to move in an osmosis event. Okay? So when we talk about water moving down its concentration gradient, we're talking about not the water in hydration shells that we call free water. The water that's not in a hydration shell. So maybe like this guy. Okay? So when you think about what direction water is going to move, a higher concentration of free water. So which side are there more hydration cells? The left side or the right side? The right side. There's a lot more bound water over here than over here. So there's more free water on this side. So the water will move from where there's a higher concentration of this free water to lower, and that's why it moves. Does that make sense? Okay. It really helps to understand the why. So you understand, like, if you don't just memorize water moves from po to per or something like that. If you understand why, you don't really have to memorize terms necessarily, okay? Now, there are terms. 38 is on the range, right? Okay. I'll just mention these words really fast, but hopefully you know because you made you do it for homework. Hypotonic and hypertonic. So use the words hypotonic and hypertonic when you're comparing two solutions to each other. I can't just have a glass of water and be like, this is hypertonic. Like, no, it's not. Hypertonic to what? To what? You're comparing two things to each other. So in this situation, this compared to this, a hypotonic solution has fewer solutes than a hypertonic solution. Hyper means like a lot. There's a lot of solutes over here compared to this. Make sense? Make sense? Okay. Isotonic is equal solute concentration. Y'all, I know you learned this in Bio 1. This is like in the standards. Do you remember this stuff?
Yeah.
Okay. Make sense? Yeah.
So on the homework, we had come here like, what is the solution of this example? Hypotonic, hypertonic, hypotonic.
Pardon this interruption.
It was like a cell with like a
Panera Bread will be in the Commons lobby at lunch today for students to check out job opportunities and fill out applications.
It's like my green job.
Wait, seriously? Mine too. JK, level
I don't think I can.
All right, we'll do more tomorrow. Y'all, if you didn't do your homework last night,
I
do
hate
1
my
through
boss,
7.
but I do.
Otherwise, just add number 7. Wait,
I like red hot sandwich.
Yeah.
Well,
I had a really chill boss over the summer. Like, I had a really chill boss and he left because he got offered a really good job. And ever since then, our new GM is like, he's actually on our old GM, was like, yeah, he sucked. He was like, nah, man, way more work than you've ever seen in your life.
work drama. That stuff I watch on get involved in.
Wait, well, like, we lost like half of like water source. Like, you have to do more. And he's a real herb. He only hires himself unless someone has a job.
So they might. Hi. Okay, I have an SNHS new member meeting. Yeah, I'll get it stopped recording. If I kick everybody out.
Like, had nothing to do one day.
Yeah, come on.
Like, hey, we'll take